Apathy is defined as reduced motivation for goal-directed behavior, and oftentimes patients with apathy don't have much insight into the condition itself, and so we really rely on caregivers to get an accurate representation of some of the behavioral manifestations of apathy. So just to name a few, apathetic behaviors include things like reduced interest in engaging in previously enjoyed activities such as work, hobbies, social interactions, as well as personal hygiene. We know that apathy is the most common neuropsychiatric symptom in Alzheimer's disease, and it's associated with a host of negative consequences. So things like increased morbidity, accelerated cognitive decline, and increased caregiver burden, just to name a few. Now, the problem is that there's no widely effective treatment for apathy in Alzheimer's disease, and this is really the driving point of this study. So what we hypothesize is that specific brain regions that have previously been implicated in apathy will interact with genetic variants in neurotransmitter systems that we know to be associated with some of the core cognitive components of apathy. So some of those components include motivation and option generation, for example. So what we hypothesize is that these regions and these genetic variants will interact to predict apathy in Alzheimer's disease. So for this study, we've pulled data from the Alzheimer's Disease Neuroimaging Initiative, or ADNI. For neuropsych data, we'll be looking at the NPI and NPI-Q to index apathy itself. We'll also be looking at some other clinical variables, so things like disease severity and cognitive ability, for example, as well as a host of different demographic variables. For imaging, we'll be looking at 1.5T or 3T T1 weighted MR images, and we'll be doing a whole brain analysis, so looking at 82 regions of interest, with our main measures here being cortical thickness and subcortical volumes. We're also interested in looking at some genetic data. Specifically, we'll look at candidate single nucleotide polymorphisms, or SNPs for short. And so these are sequences in the DNA where there's been a single base substitution, which gives rise to variable genotypes in the population and, as a result, different phenotypes. For our data analysis, we'll in the future be looking to do a partial least squares correspondence analysis. But in the meantime, I'll show you some preliminary results from a principal component analysis and binary logistic regression that I've done with the imaging variables. So the overall prediction of the study is that specific regions known to be implicated in apathy will interact with these SNPs in the neurotransmitter systems we know to be associated with the cognitive components of apathy to predict apathy in our disease cohort. So over here, just some preliminary results from the principal component analysis. We found that there were several brain regions that do largely overlap with some of the regions known to be associated with apathy to be predictors in our logistic model. And there were also some clinical predictors as well, including the clinical dementia rating scale, so disease severity, as well as years of education. So to summarize, apathy is a debilitating symptom of Alzheimer's disease, and currently there's really no widely effective treatment for it. But we think that perhaps there'll be some interactions between specific brain regions as well as genetic variants, so SNPs in particular, in neurotransmitter systems that are associated with the core cognitive components of apathy. And these may help us predict whether apathy will occur in an Alzheimer's disease cohort. And the results of the study may help to inform future pharmacological treatments in clinical trials for apathy. Thanks for listening.